Genetics, Lesson 2.8 Review. So again, this is just going to be a quick review of all the things that we've learned in this unit. And maybe if you just want to watch this quickly before you have a test, or if you just want to review some quick concepts to get a quick understanding of them. So a lot in this unit we talked about, of course, DNA, and we talked about genetics, and we talked about different alleles and traits that make us essentially who we are. We talked about reproduction, and we talked about growth and repair in mitosis, and all of those things connect and are essentially the basis for life and the basis for how humans, and not only humans, animals and all organisms exist. So the first lesson we talked about is DNA. But what is DNA? So also known as deoxyribonucleic acid. It's a twisted ladder that kind of looks something like this. So if you ever picture DNA, it would look something like this, sort of ish. And it's double-stranded, and which means that it has two strands, and each strand consists of a chain of units called nucleotides. DNA is anti-parallel, so that means if one strand, like down here, starts with a 5 and ends with a 3, its corresponding strand is going to start with a 3 and end with a 5, so just the exact opposite. And then each nucleotide has 3 parts, so a phosphate group, the sugars, and then the nitrogen bases, so the A, the T, the C, and the G. And the name for the A is adenine, the name for the C is cysteine, and then the name for the G is guanine, and then T is thymine. So we always remember that the A's always go with the T's always go with A's, and then the C's always go with G's, and G's always go with C's. So just like I have written over here, if this was the first strand that I gave you, this red one, what would be the corresponding strand? So again, it starts with 5, so we start the other one with 3, ends with 3, start the other one, and the other one with 5. And then since A goes with T, C goes with G, T goes with A, G goes with C, G goes with C, A goes with T, C goes with G. And those are pretty simple to figure out. In the next lesson, we talked about mitosis, and remember that mitosis is for growth and repair. So we have all these different phases, and we start off with interphase, which is when the cell replicates its DNA and individual chromosomes are not yet visible. The presence of there is a presence of the nucleus, and then this indicates that the cell is making proteins. Then we have prophase, where the sister chromatids become visible, so those little purple bits in the middle, and they attach to the long fibers at the end of the spindle. And then the nucleus disappears, and the nuclear envelope starts to break down, so that sort of dotted line around starts to break down. The next phase that we have is metaphase, is when the sister chromatids are fully attached to the spindle fibers, and the spindle fibers move the chromatids and they line them across the middle of the cell. Next, we have anaphase, which is when the sister chromatids separate almost fully, and they start to form individual partners to become by themselves. And then once the chromosomes have reached the poles, so remember each of those opposite ends of the cell are the poles, and then they start to form their own separate groups. In telophase, the chromosomes begin to uncoil and the spindle fibers disappear. The nuclear envelope reforms around each, and they start to form their own 2 cells. And then we have cytokinesis, which remember we say happens at pretty much the exact same time as telophase, and that completes the cell division, and it divides the cytoplasm in the daughter cells. So that's essentially mitosis. Next, we have meiosis, and meiosis is a bit more complicated because there is lots of different phases. So remember, we have meiosis 1, where the homologous chromosomes separate, and then we have meiosis 2, where the sister chromatids separate. So just a quick overview. So in prophase 1, so that purple part up there, just like in mitosis, the chromosomes condense and the nuclear membrane dissolves. This is what's known as synapsis, and each of them forms a tetrad. So remember that cross shape that kind of looks like this is what's known as a tetrad. Then in metaphase 1, the homologous chromosomes in pairs move to the equator of the cell, so the center of the cell. And then once each chromosome is replicated and the replicated chromosomes are joined by the centromere to which the spindle fibers attach, the homologous chromosomes are attached at their chiasmata, which holds the pair together at the metaphase plate. In anaphase 1, the homologous chromosomes are each pulled by their centromere to the opposite poles of the cell, so again those top 2 points, but the sister chromatids remain attached. And then in telophase and cytokinesis, the homologs are now separate and each pole is now haploid. The result is 2 haploid daughter cells, each with half the number of chromosomes of the parent cell. Next, we sort of go down into that part that's turquoise again, and we're kind of put a question mark beside it because some cells don't go through interphase after meiosis 1, some cells go through a very short one, some go through a very long one, but in any event, there's no replication of DNA here. In prophase 2, the spindle fibers begin to appear again, and they attach to the chromosomes at their centromere and begin to move to the metaphase plate. In metaphase 2, the chromosomes line up at the metaphase plate or the equator, just like in mitosis. In anaphase 2, the centromeres of the sister chromatids are fully separate now, and the sister chromatids of each pair are now independent chromosomes. Daughter chromosomes also move towards the opposite poles. And then lastly, telophase and cytokinesis. Remember, they happen at almost the same time. Nuclei begin to form at opposite poles and the chromosomes unwind. The end result is the entire process is 4 daughter cells with haploid number of chromosomes. So that's essentially what meiosis is. We also talked about in the next lesson Mendelian genetics. So remember, this is when he did the experiment with determining the different flowers, and he did with the pea shapes. So essentially what he did is he took purebred things like this pure purple flower and this pure white flower, and it gave him all purple flowers in the next generation. Well, he thought to himself, well, how's that possible? If I had a white flower, how does that give me all purple flowers, even if I mix them together? Well, he realized that the purple one was more dominant, so it told all the other ones to be purple. In the next generation, when he crossed the purple flowers together, he actually got 3 purple and 1 white. He thought to himself, well, how is it possible that I had 1 white flower? Well, it's because the white flower from the original generation, the parent generation, it was the recessive gene. So in the F1 generation, which was this one, all of these guys carried the recessive gene of white in order for this guy to be white. And this is just a little bit of review from the traits and alleles. So remember that homozygous dominant, for dominant things we use capital letters, and homozygous means same, so it's going to be capital capital or big P big P. For homozygous recessive, again, it's the same except just for the recessive, and for recessive we use lowercase, so it's going to be little p little p. And then the last one is heterozygous, which means like different, so it's going to be big P little p. Next, we talked about sex-linked diseases. So here are just some quick notes on it, but all you need to basically know is that they're traits whose phenotypic expression is controlled by the genes found at different loci. And then we talked a little bit about autosomal traits and diseases, which are carried on any of the chromosomes that do not determine gender, and then sex-linked traits and diseases, which are traits of disease carried on the X or the Y chromosomes. So remember that we represent girls as XX and boys as XY. We also then talked about pedigrees, and we talked about how to draw them and how to differentiate the different traits that could be carried if it's autosomal recessive or autosomal dominant or sex-linked recessive or sex-linked dominant. Okay, we're at the end of this video, and now you're just going to pause the video here, and then I will show you the answers on the next slide. But try to do these ones by yourself, and then if you need help, you can always look at the answers. Okay, so here are the answers for you to take a look at. Most of them are pretty straightforward, and you can always go back in the lessons to see a further explanation of them. And that's it. We are finished Unit 2 of Genetics.